All right, we are barely going to dip our toe on the pool here on this topic. So we're just going to mention a couple ways that genes are regulated. This is so much more complicated than we're putting this out here for you, but we're just trying to get you a little bit introduced to the fact that genes are not all on, they're not all off. There are signals to turn genes on or off. They're what we call inducible genes, which we can turn on, or repressed genes that we can turn off. And so we're just going to kind of talk about, just like I said, just a little bit about gene expression. So again, when we talk about gene expression, we're talking about making that messenger RNA. If there's no messenger RNA being made, then you don't make the protein and the protein is not expressed. And so we only need a small amount of DNA that's expressed at any given time. So most cells are very specialized in what they do. So they may only have a few genes turned on. So the cell will regulate that expression on what they need. So we always talk about in prokaryotes, we always talk about this lac operon, and this is actually in E. coli. All right. And so with the lac operon, what happens is the enzyme lactose. Now, you do not need the enzyme lactose unless you have lactose available. So you don't need lactase, sorry. The enzyme is lactase. We don't need lactase unless we have milk sugars. All right. So why would we make an enzyme if we don't have that product to do anything with it? So what we have here is, again, remember I talked about the promoter. The promoter is the place that we will say, hey, RNA polymerase, this is where you're going to join. This is where we're going to start. This is the beginning of the gene. Well, we have in here between the promoter and our actual instructions for our messenger RNA, what's called an operator. And the operator is going to help control whether or not that gene is on or off. In this particular gene, it's repressed. We have a repressor here, and so my RNA polymerase cannot make a copy. It's being blocked. If we have lactose, okay, so if we have that milk sugar, it binds to the repressor, and the repressor, so it would bind here, and then the repressor falls off. So it's saying, yep, we have lactose, we need you. And so once that falls off, now our RNA polymerase can continue to make the protein lactase, which will break down the lactose. Once lactose broke down, guess what? Our repressor will go back and bind to it because we'll have broken down the lactose. And so that is what we would call an inducible gene, in that we could turn it on. It is repressed unless we turn it on. Another way that we can control gene expression in eukaryotes is that we can actually have our DNA wound up super, super tight. And so if our DNA is wound up tight, it's like, if your book is closed, you cannot read it. And so we have these proteins called histones, and we have these tails, and this is a whole area of what we call epigenetics now. And so notice that we have to unwrap. So we have this area of DNA where we can read those instructions. And so we have what's called these little tails, and some are methylated and some are acylated. And based on whether they're acylated or methylated, they will either be wrapped up tight or they will open up so we can read the gene. So that helps control gene expression. And that is all we're going to talk about with gene expression. So just kind of gave you a little bit, one mechanism of gene expression in prokaryotes, and then one mechanism, there are many, for eukaryotic cells. And so then we have our chapter conclusion, and that is it for chapter 10. Hopefully you can try to get this done as soon as possible. So you'll have your 8 and 10 done, but we do need to start moving on and doing some genetics, so we're prepared for our next lab. So I will be starting to work on your chapter 9 material, which is going to take several weeks. We'll be working on that one for 2 to 3 weeks, just that one chapter. Right. So hopefully if you have any questions, hopefully this was helpful. And if you have any questions, please feel free to give me a shout out and I will see you in the next video.